Hello, all. Welcome back from your lunch. I hope it was great. I hope you enjoyed the videos on the compilation videos from Sam's talks and from people speaking about Sam. So again, you may recognize me from earlier. I'm Bryan Smith, an associate professor in biomedical engineering at Michigan State, where we focus on harnessing the power of the immune system to control and track immune cell fates. And so it's my pleasure now to introduce Julie Sutcliffe, Dr. Julie Sutcliffe. Julie is a professor of internal medicine and biomedical engineering at UC Davis, University of California, Davis. She's the co-director of the Center for Molecular and Genomic Medicine, and also the director of radiochemistry. She, as befitting her research group, her translational lab group, she's also the PI of 3 active clinical trials. She's the PI of the UC Davis U01 Pancreatic Cancer Detection Consortium, and she leads the UC Davis Lustgarten Foundation Stand Up to Cancer Pancreatic Cancer Collective Research Team. Dr. Sutcliffe is a fellow of the Society of Nuclear Medicine and Molecular Imaging. She's a fellow of the World Molecular Imaging Society, or WMIS, and she's a fellow of the American Institute for Medical and Biological Engineering. She's now the co-president of the World Molecular Imaging Society, and I'm now pleased to welcome the audience, to a pre-recorded seminar of Dr. Sutcliffe due to a time conflict. So unfortunately, there won't be any availability for question and answer after this talk. But I'm happy to introduce Dr. Sutcliffe to discuss her molecular imaging, in particular imaging integrins, bench to bedside. Thank you.
Thank you, Dr. Smith, for the kind introduction, and thank you to Drs. Wu and Gold for the invitation to speak today. So like to many of you joining today, Sam was a mentor and colleague to me, even though we never actually worked together at the same institution. I was honored to serve on the Cancer Prevention Research in Texas study section that Sam chaired. He always made us feel that we'd done our due diligence on every single grant that was submitted. He kept us up to date with his knowledge on the latest and greatest technologies that we all hoped would help our patients. And he actually made study section fun. So today I'll be talking about some of our work on targeting integrins, an area that Sam and his team also invested in. So these are my disclosures, and relevant to today's talk, I'm the founder and CEO of Luminance Biosciences and hold intellectual property on several of the compounds that I will be talking about today. So the integrin alpha V beta 6 is an epithelial specific cell surface receptor. It's low or undetectable on most healthy adult epithelium. It is overexpressed on a wide variety of cancers. It plays a key role in invasion and metastasis, and its expression is often associated with poor prognosis for the patients. So based on the high contrast between malignant and healthy tissue and the functional relevance of this integrin, we and others believe that it's an important target for the development of both imaging and therapeutic compounds for a wide range of carcinomas, and including some of the most lethal malignancies, such as pancreatic ductal adenocarcinoma. In fact, alpha V beta 6 was first identified in pancreas cancer. So our approach towards imaging the alpha V beta 6 receptor has been to use peptides. So why peptides? Well, peptides are relatively small in size, can be relatively easily synthesized, and can be modified significantly for labeling and to improve their in vivo stability. Now, they can be identified in several ways, including rational approaches and random approaches. So the rational approach is used when we have a lot of information about the target. We might know its binding ligand, for example, or we might have its crystal structure. Now, in cases when we don't know anything about the receptor, or its ligand. We often use combinatorial approaches, some more random approaches, such as phage display and the 1-bead-1 compound. Now, unfortunately, when we started research to develop agents for beta-6, very little was known. The non-binding ligand was not known, and we did not have a crystal structure available to us to enable a rational design. So where did we start? Well, there was actually numerous reports in the literature describing the role of the integrin alpha beta 6 in the foot-and-mouth disease virus. So you're probably wondering, well, what does that have to do with imaging the integrin in cancer? Well, the foot-and-mouth disease virus enters its host cell via the integrin alpha beta 6, and the crystal structure of the virus and the binding domain was known. And it became apparent over time that a 20 amino acid sequence that I'm going to refer to moving forward as A20FMDB2 that was found on the GH loop of the viral protein 1 was responsible for the virus getting into its host cell. So this 20 amino acid sequence was the foundation of our first generation compound. So we took this 20 amino acid sequence from the virus and made our first fluorine 18 peptide. So I'm including this slide here for the chemists in the audience. And here we see the process involved in synthesizing our first F18 peptide. So we start with the production of fluoride in a cyclotron in Curie amounts, and through the fluorination of a triflate precursor, we make fluorobenzoic acid. This fluorobenzoic acid is then coupled onto a peptide, and then we have to purify the product using HPLC. Now, you have to remember that all this work is radioactive, so it has to be done remotely using robotic manipulators. And here you can see Dr. Hausner demonstrating his radiochemistry skills. So of note, the overall process here, including final quality assurance, is approximately 3 hours. And just as a reminder, the half-life of fluorine 18 is 110 minutes. So next, the compound was taken in vivo into a mouse model. So our mouse model has a set of paired xenograft tumors. So what do I mean by that? So on the right flank of the mouse is the DX3 puro xenograft. Now, DX3 puro is a melanoma cell line that naturally expresses many of the integrins such as alpha B beta 3, alpha B beta 5, alpha 2B beta 3. And then on the left flank of the animal, we have a DX3 puro beta 6 xenograft. Now, this is the DX3 puro base parent cell line that's transfected to express the alpha beta 6 integrin. So the only difference between the two xenografts is our integrin of interest. So we really think this is a very stringent model. So the images you see here, the images on the right are transactional images through the tumor showing uptake of our first F18 peptide. And you see uptake only in the alpha beta 6 expressing tumor. Now, the images on the left are images of the animal after injection of FDG, so the gold standard PET imaging agent in cancer. And as chemists developing novel PET agents, we always have to compare our compounds to FDG, because everyone's saying, well, FDG is so great, why is your compound any better? So we use FDG as our control, and in this case you can see our peptide is only in the beta 6 positive cells, whereas you see FDG uptake in both tumor cells. So we're saying this is a very alpha beta 6 specific peptide. Now, the animals were then euthanized and radioactivity in all the organs counted. And although we could see contrast in the positive tumor, the agent was rapidly cleared out of the kidneys and rapidly cleared out of the tumor. So I think in conclusion, and this was the conclusion in our first cancer research paper in 2007, our conclusion was that although F18 fluorobenzoic acid A20-FMDB2 was successful, we anticipate that modifications of this peptide to improve biological stability whilst maintaining specificity will result in improved targeting imaging agents. I just don't think we had truly anticipated how many modifications would be necessary and how long it would actually take us. So over the next several years, my team made a series of modifications to this peptide to improve its pharmacokinetics, but also to simplify the chemistry. And we are now at generation number 5. So this peptide, it has nanomolar affinity for the integrin alpha beta 6 with low or pretty much zero affinity to the other integrins such as alpha beta 3 and alpha beta 5 as indicated by ELISA. Now in cell studies, the compound demonstrates rapid and selective binding and internalization into alpha beta 6 positive cells. And the peptides demonstrated favorable tumor targeting and clearance in numerous mouse models. It's predominantly excreted through the kidneys. And finally, we have demonstrated favorable pharmacokinetics in non-human primates at an imaging resource we're fortunate enough to have here at UC Davis, allowing us to actually screen our compounds in non-human primates prior to transitioning towards clinical studies. So this peptide is now translated into the clinic, and our first-in-human study is almost complete. Patients with a diagnosis of breast, colon, lung, or pancreas cancer were eligible for the study. To date, we've scanned 26 patients. These patients are injected with 10 molecules of the peptide and imaged using PET-CT at multiple time points. Now, as this is a first-in-human study, obviously there's a lot of safety things we have to look at. So we have multiple blood draws are taken throughout the study, EKGs and vitals are monitored throughout the study, and on day 1 and day 7 following injection of the radiotracer. But it's to say that the peptide was well tolerated by all patients. So I'm going to show you just a few images here. So the images here on the left are images taken after injection of the peptide in a patient with metastatic lung cancer. So here you see the maximum intensity projected image, the MIP image after injection. And this is 1 hour after injection of the tracer. And you can see uptake in the primary lung lesion as well as in bone metastasis. And the peptide is renally excreted as we saw in our preclinical studies. Here is just a transaxial slice through the primary tumor, and you can see on the CT and the corresponding uptake of our compound in the primary lung tumor. Now, unfortunately, there was no tissue biopsy available for this patient, but we did have an FNA sample of a pleural effusion, so we labeled that sample with a beta-6 antibody for immunohistochemistry, and you can see that the tumor cells in the sample were positive for alpha beta 6. Now this image here is a brain image of a patient with non-small cell lung cancer who had known brain metastasis, and this is a fused PET MR image taken after injection of our compound. This is at the 1 hour time point, and we can see uptake of our compound in sub-centimeter lesions in the brain. And finally, this image here is our images taken of a patient with a suspect recurrence metastatic breast cancer post-bilateral mastectomy. The image on the right being the MIP image after injection with our peptide, and the image on the left being the FDG PET image. These images were acquired 1 week apart, with the peptide images being acquired first. Now you can see numerous lymph nodes and bony mets in the image of our peptide. And of course you can see several of them with FDG PET, but our agent was a lot more sensitive and saw a lot more lesions than the standard of care FDG PET. So I think to summarize the first in human data to date, the peptide is well tolerated in all patients. It's predominantly renally excreted, as was observed in our preclinical studies. The PET images demonstrated significant uptake of our F18 peptide in both the primary lesion as well as metastasis, including sub-centimeter lesions in adrenal glands, bone mets, brain mets, liver mets, and lung mets. So this is all great, but these patients already knew that they had metastasis. So what can we now do with this information to help these patients? How do we transition quickly from a diagnostic to a therapeutic? Well, we use a theranostic approach, i.e. we combine therapy and diagnostics, often using the same molecule. So the radiotheranostic strategy is not new to nuclear medicine. We've been using this approach for decades now, for example, using radioactive iodine to both detect and treat thyroid cancer. So the principle here is we're going to use the imaging agent to diagnose and select patients. We're then going to treat the patient with the radioligand, for example, using a peptide receptor radionuclide therapy. And then we're going to re-image them to monitor treatment response. So here on the left, you see an image of a patient with a neuroendocrine tumor that was imaged with NetSpot, a peptide radiolabeled with gallium-68. They were then treated with 3 cycles of the peptide receptor radionuclide therapy, Lutathera, and they were re-imaged after the last treatment. And you can see in the post-treatment gallium image a significant reduction in disease burden. So we're now proposing to leverage this kind of technology and using a similar approach where we're going to look at the peptide we've discussed, radiolabeled with radiolabeled with gallium-68, the positron emitter, 68-minute half-life, to identify patients that would be suitable for treatment with the lutetium labeled peptide, lutetium-177, beta minus emitter, a radionuclide with a 6.73 day half-life. So we've now synthesized 2 new peptides. Peptide 1 was radiolabeled with gallium-68. Peptide 2 was appended with an albumin binding moiety to increase its circulation time and then was labeled with lutetium-177. Now, both constructs demonstrated good serum stability. So the gallium construct was stable over 2-hour time point, and lutetium-177 over 24 hours. Both constructs demonstrated selective binding and internalization into alpha beta-6 expressing cells, and the binding and the internalization correlated to levels of expression of the integrin alpha beta-6. So for example, KPAN2 demonstrated high cell binding and high expression of beta-6, and PANK1 had low cell binding and had 0 expression of alpha beta-6. So the cell toxicity of the lutetium construct was then evaluated in several assays, including WST1, caspase-37, and a clonogenic assay. And what we saw was by increasing the amount of radioactivity of the lutetium construct resulted in a decrease in proliferation, an increase in apoptosis, and a decrease in overall survival. So to summarize the in vitro testing, both constructs demonstrated high purity, both demonstrated good stability and alpha beta 6 dependent cell uptake, and the lutetium construct demonstrated cell toxicity. So these constructs were then evaluated in vivo in several preclinical models, but here I'm just going to show you some of the data in a BXPC3 xenograft model. The gallium-68 was imaged over a 2-hour window, and ex vivo biodistribution was performed at 1 and 2 hours. So the transaxial and coronal sections are shown here after injection of the gallium construct. It was predominantly renally excreted and the beta-6 positive tumors visualized. The ex vivo cut and count BioD data corresponded to the imaging data with clearance from most organs and tissues over time, but some retention was noted in the tumor and some in the stomach and small intestine. Now, the lutetium constructs with and without albumin binding moiety were evaluated in vivo, and it should be noted that the construct containing the albumin binding moiety shows superior pharmacokinetics. It had almost 5 times the amount of uptake in the tumor, with clearance from other major organs such as stomach and kidney over time. You see clearance the kidney here, the stomach over time, but retention in the tumor. So overall, this resulted in improved tumor to non-tumor organ ratios. These biodistribution data were then extrapolated to estimate the human dosimetry data using a Linda. We found that the major dose-limiting organ based on the murine data are obviously the kidneys and the stomach wall, with doses calculated at 2.27 and 0.2 millisieverts per becquerel respectively. Based on our plans for the first in-human study with the starting patient dose at 25 millicuries, we estimate that the dose to kidneys would be 2 gray, so much lower than the renal dose limit of 23 gray. We've also performed several urine therapeutic efficacy studies, and here I'm showing the results in our paired model. We monitored mice for body weight, we monitored CBC and CMP, and we didn't see any changes. All our control animals did reach endpoint, i.e. their tumors grew greater than 2 centimeters in 69 days, and the treated animals with a positive tumor did not reach endpoint within the 120 days of the experiment. So to summarize, the estimated human dose is well below the maximum allowable limits. It's comparable to other lutetium compounds that are currently being used as therapeutics, and we showed promising therapeutic efficacy in mice. So to wrap up, because I know I'm already way over time, I wanted to share some of our preliminary data for our first in-human study using this new theranostic pair. The overall goal of the study is to evaluate the safety and efficacy of the theranostic pair in patients with locally advanced or metastatic pancreas cancer, and it's a classical 3+3 study design. We are using the gallium construct to identify patients that are eligible for treatment. So those patients who have uptake in lesions 2 times normal lung or 2 times normal liver are deemed eligible for the study. The starting dose for lutetium being 25 millicuries, and we're stepping up to a maximum of 200 millicuries. So the image here on the left, you can see is a patient. This is a patient who had Whipple surgery and had multiple treatments with chemotherapy, and then unfortunately presented with liver metastasis. And you can see here uptake of our construct in many of the liver lesions. SUV values are 15 and above. We also did see some additional lung lesions and bony lesions that were not previously detected. This patient did go on to receive 150 millicuries of the lutetium construct and was imaged with SPECT the day later. And you can see that the construct is being renally excreted, but we do have drug on target. So to summarize, I think we can all agree that molecular imaging with molecularly targeted probes plays a critical role in patient care. I hope I've convinced you that alpha v beta 6 is a promising target for both imaging and treatment, and that our peptide is a promising theranostic. It's been a long journey from our first preclinical imaging study in 2007 to our first treatment study in 2021. But if I can leave you, and in particular our trainees in the audience, with one last take-home message, please, please, please be persistent. To be able to see your compounds translated from the bench into something that can benefit our patients is truly rewarding. So with that, I'd like to thank all of those who have supported these efforts. It really, really does take a village. My lab, who continue to work tirelessly through this pandemic. The imaging team at the Center for Molecular and Genomic Imaging, staff at the Primate Center, the nuclear medicine staff, and my clinical supporters, Drs. Bold, Chu, Daly, Foster, and Kim, and obviously the funding agencies. Without their support, we couldn't have done a lot of the work I presented today. But I want to give a special thank you to our patients and their loved ones for their trust and support in our research. After all, they are the reason that we go to the lab every single day. So with that, I thank you for your attention. I'd like to thank the organisers again for the opportunity to share our journey with you today. I'm really sorry I can't be there for the streamed Q&A session, but please, please, please don't hesitate to reach out via email or Twitter if you have any comments or questions. Take care, everyone.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.